So in my last videos, we've explored lots of different agonists and antagonists and how they can bind to different sites such as orthosteric and allosteric sites to produce a variety of responses. But we only focused very briefly on antagonists. So in reality, antagonism is a complex issue with lots of different types and variations of antagonists. This video is fully dedicated to antagonism. We're going to learn about a bunch of different types and we're going to study the differences between all of them. Let's go. Now before we get ahead of ourselves, I just want to say I'd highly recommend watching my past videos on allosteric and orthosteric sites and my other video about partial, inverse, and full agonists as well as antagonists. These videos will give you some foundational knowledge that will really help understand the more complex concepts in this video. So I've left the links for both of these past videos down in the description below, so be sure to check them out. So the first way that we're going to differ between types of antagonism is pharmacological versus physiological antagonism. So this is definitely the biggest distinction we can make when looking at antagonism. Physiological antagonism involves two separate receptors, while pharmacological antagonism involves just one type of receptor. So two distinct types of receptors, just one receptor type. So how does this work? Let's first take a look at physiological antagonism. So as we said, physiological antagonism, it involves two separate types of receptors. As you can see here, we have receptor type A and receptor type B. So both of these receptors are linked to two separate responses. So when messenger molecules, such as, let's draw our different messenger molecules. You got this one, and then you got this one. So your messenger molecules, they come and bind to your two receptors, and they produce responses inside your cell. But here's a catch. Your two responses, they sort of counter out each other. They act against each other, creating a fine balance between the two responses produced by your two separate receptors. And because these two responses are countering each other out, this creates a sort of antagonism. What's unusual about physiological antagonism is that it actually does not require an antagonist. Because think about it. You have two agonists binding onto two receptors to both produce their designated responses. So there's no antagonists involved, only agonists and receptors. But the two responses produced by these receptors, they counter each other out. So let's say this one is an increase in contraction and this response is a decrease in contraction. These two responses act against each other, creating a fine balance. And this type of antagonism is very useful because it helps prevent responses from getting out of control. It keeps response levels in check. And if response levels get out of control, this could be very dangerous for the body. So this type of antagonism is essential. Now, it's important to note that the agonists that are binding onto your receptors, they can be two completely separate agonists, or they can actually both be the same type of agonist. So let's look at an example of this. So the agonist that's binding onto your receptors and creating physiological antagonism, it can either be the same agonist binding to both receptors, or it can be two separate agonists, each binding to its own receptor. But the concept is the same. So for example, histamine is one agonist that can bind to both, it can bind to both H2 and H3 receptors. Now H2 receptors, they increase cAMP levels while H3 receptors decrease cAMP levels. So these two responses can sort of counter each other out, creating a balance of physiological antagonism. As you can see, it's one agonist binding to two receptor types. On the other hand, in airway smooth muscle tissues, you can have norepinephrine, a messenger molecule, come and bind to its own receptors, and you can have acetylcholine, which is another messenger molecule, come and bind to its own receptors. And norepinephrine receptors cause the response of relaxation, like relaxing smooth muscle, whereas acetylcholine causes the smooth muscle to contract. So the relaxation and contraction of smooth muscle, these two counteract each other, creating a balance, they also create a balance of physiological antagonism to help regulate airway smooth muscle contraction and dilation. This is why when you're having an allergic reaction, you take an EpiPen. Epi is short for epinephrine, so that it opens up your airways. It dilates and relaxes your airway, allowing you to breathe easier and survive the allergic reaction. By the way, if you want to learn more about histamine or norepinephrine or acetylcholine, I've made videos for all of these. Be sure to check them out. They're in the description below. But now you can see how you can either have two different agonists binding onto two receptor types to create physiological antagonism, or you can have just one agonist binding onto two different types of receptors to create physiological antagonism. Okay, so that's physiological antagonism, which involves two separate types of receptors. On the other hand, pharmacological antagonism involves just one receptor type. And a pharmacological antagonism can be broken down into competitive versus non-competitive antagonism. Now remember what we learned in our last video about orthosteric and allosteric sites. So you have a cell with a receptor, and receptors can have different sites. Your orthosteric site, which is like your main site, that's where agonists can bind to produce responses inside the cell. But your receptor can also have this secondary allosteric site, which is like a regulatory site, where modulators can come and bind to regulate and fine-tune responses. Understanding these two distinct sites, orthosteric and allosteric, is crucial for understanding these next types of antagonism, competitive and non-competitive, which are both types of pharmacological antagonism. So competitive antagonism is when an agonist and an antagonist compete to bind onto the same orthosteric site. So remember, the orthosteric site is where agonists can come and bind onto. I'll represent the agonist with this little blue circle. It's where agonists can come and bind onto, and when they do, they change the conformation of your receptor, and that produces an intracellular response. But the thing is, antagonists, they also come and bind onto this same orthosteric site. So just to know, I'm using blue to represent agonist, and I'm using red to represent antagonist. So antagonists, they come and bind to the same orthosteric site, but when an antagonist binds onto it, it doesn't cause a change in conformation. It just sits at the site. So I'm going to draw the antagonist sitting inside your orthosteric receptor. It doesn't cause a conformation change, and it doesn't cause any type of response inside the cell. It just sits there. But the effect of that is that now your agonist is unable to bind because this antagonist is in the way. It just blocks agonists from binding onto it. So let's say first you have a bunch of agonists. So I'll demonstrate that with a bunch of blue circles, right? These agonist molecules are going to be moving around randomly, and they are going to be coming and binding onto your receptors, producing responses, and then after some time they're going to leave your receptors. So at the start you just have agonist, but then all of a sudden you also add some antagonists. So now this antagonist is going to be competing with your agonist because they're both competing to bind onto the same site. Hence the name competitive antagonism. So the effect of this is that your agonist is going to have a harder time binding to these receptors because it's now competing against something else. Let's say your antagonist gets there first and it comes and binds to your orthosteric receptor. Now it's blocked. So your agonist cannot bind onto it until this antagonist leaves, and then the site is free again for the agonist to come and bind. So they have to take turns, they have to compete against each other. So this lowers the response that is produced compared to if you just had agonist alone without any antagonist. If you want a more in-depth explanation about this type of antagonist, be sure to check out my video about partial, inverse, and full agonists and antagonists. I've left a link of this video down in the description below. So then what's a non-competitive antagonist? Well, if competitive antagonists compete to bind onto the same site, then non-competitive antagonists must be the opposite, right? They must bind onto separate sites. And that's exactly what happens. In non-competitive antagonism, your agonist still binds to your orthosteric site, but your antagonist actually binds instead to your allosteric sites. They bind onto separate sites. And this means that your antagonist can also be called a negative allosteric modulator. Now, if you want to learn more about allosteric modulation, be sure to check out my last video about allosteric and orthosteric sites. I'll link it down in the description below. But basically, yeah, your negative allosteric modulator binds to the separate allosteric site, and in doing so, it lowers or antagonizes the response that your agonist normally produces. And it usually does this by, when it binds to your allosteric site, it causes a conformational change in your receptor that maybe it lowers the receptiveness, or maybe it makes it less effective at producing responses inside the cell, but in some way it's able to suppress or antagonize the response that your agonist is normally able to produce. So the key difference is that your agonist and your antagonist, or negative allosteric modulator, are binding onto two completely separate sites, and because of that, they're not competing against each other. Whereas with competitive antagonism, only one thing can bind to a site at a time, either your agonist or your antagonist. In this case, your agonist can be bound to your orthosteric site, and your antagonist can be bound to your allosteric site at the same time. So they're not competing to bind onto the same site, hence the name non-competitive. Okay, so now that we've looked at competitive versus non-competitive antagonists, so your agonist and antagonist binding onto the same site versus binding onto different sites, the last way which we can break down competitive antagonism even further is by comparing surmountable versus insurmountable antagonism. So basically, surmountable antagonism means that you can overcome this type of antagonism by adding more an agonist, while insurmountable means that the antagonism is insurmountable. No matter how much agonist you add, you cannot overcome or surmount the antagonism that is taking place. So let's take a look at what this might look like. So remember, with competitive antagonism, we have both agonists and antagonists which are competing to bind onto the same orthosteric sites. And as you can see here, we have a cell with a bunch of receptors. I'm being lazy and I'm only drawing the orthosteric sites and not the allosteric sites, because we don't really need to focus on allosteric sites for now, because remember, with competitive antagonism, both your agonists and antagonists are competing to bind onto the same orthosteric sites. So we have a bunch of agonists, drawing those in green, and we have a bunch of antagonists. And I drew them separated, but in reality they're all, mixed up together. They're all floating around together. And your agonists and antagonists will move around randomly, or we call this stochastically. So stochastic movement is the random movement of your agonists and antagonists around around the fluid, around the extracellular space, the space outside the cell. And as they move around randomly in all directions, they may, just by chance, they may happen to bind onto a receptor. So let's say this agonist comes and binds onto the receptor, right? So once this agonist is bound, they may leave after some time, and then they can continue to move around. And then after it leaves, then this receptor is free. So maybe an antagonist can come and bind to it, or maybe an agonist can come and bind to it. So it's a constant process of agonists and antagonists moving around in all directions, binding onto receptors, leaving receptors, and taking each other's places. So this creates a balance of stochastic movements with your agonists and antagonists. So because everything's moving around randomly and the process of binding onto receptors is up to chance, the greater concentration you have of something, the greater chance it has to bind onto receptors. So let's say you have a high concentration of agonist, then there's a higher chance of these agonists binding onto your receptors and producing responses. But then let's say you start adding in some antagonist. Now these antagonists are also going to be floating around, moving around stochastically, and they'll have the possibility of binding onto some of these receptors. So now some of your receptors may be occupied with antagonists, meaning that agonists won't be able to bind onto these receptors because they're already occupied, and therefore your response levels decrease. And as you increase the concentration of antagonists, adding more and more and more and more, the chance of your antagonists binding becomes higher and higher, while the chance of your agonists binding becomes lower and lower. So therefore, the more antagonist you add, the more cell response levels continue to decrease. So surmountable antagonism is when your antagonist can bind onto orthosteric sites, but then after some time it can leave. So it can go and bind, and then after some time it can unbind as it moves around stochastically. And when it unbinds, this means that agonists are now able to come and bind to this receptor, because after it leaves, this receptor is vacant. So these agonists, which are moving around stochastically, may be able to come and bind onto those receptors. So for this type of antagonism, if you add a lot of agonists, so let's say we add a bunch of agonists, right? So there's a higher concentration of agonists compared to antagonists. If you do this, then the higher concentration of agonists can basically outcompete the agonists. And this is because a higher concentration of agonists means it's more likely for agonists to bind to these orthosteric sites instead of antagonists. And this makes sense, because remember, like we said, because everything is moving around stochastically, randomly, the higher concentration you have of something, the higher chance it has to bind. So, more agonist concentration means it's more likely for these agonists to bind. And even if some antagonists do end up binding to receptors, after some time they may leave, and then they are likely to be replaced with agonists because, like we said, there's such a high concentration of agonists compared to antagonists. So that's why this type of antagonism is called surmountable, because it can be overcome or surmounted by adding a lot of agonists, by adding a large concentration of agonists. So if we look at dose-response curves for this type of antagonism, they would look something like this. So this first curve, the green one, this represents when you add agonist without your antagonist. So as you add agonist, as your agonist concentration increases, response slowly increases, and then it increases faster and faster, and it spikes up, and then it plateaus at the maximum possible response. That's without an antagonist. Now, when we add an antagonist, we need a higher concentration of agonist in order to overcome or surmount this antagonist and produce the same responses. So as you can see, we're able to get the same level of response, just we need a much higher concentration. I should extend this. We need a higher concentration of agonist to do this, because remember, we need a higher concentration of agonist to outcompete the antagonist which we have now added. So as a result, your curve is basically shifted to the right. And this results in a decrease in potency because you need a higher concentration to achieve the same results. So then what's insurmountable antagonism? Well, let's say if your antagonist binds to your orthosteric sites, but it sticks permanently. It cannot leave, meaning that an agonist cannot ever come and bind to this orthosteric site. So we can think of this orthosteric site as being permanently disabled. There's no way for agonists to bind to this site no matter how much you add, because it's permanently occupied by this antagonist. The receptor is basically gone. That's why this antagonist is insurmountable. It cannot be overcome or surmounted no matter how much extra agonist that you add. So let's look at this entire cell, right? We have 10 orthosteric sites, and we have a concentration of agonist that can go and bind onto these orthosteric sites and produce response. But now we're going to add some of our insurmountable antagonist. So let's say this insurmountable antagonist goes over and binds to 4 out of our 10 receptors. I'm just going to delete these arrows to make it a bit clearer. But now, so basically, out of the 10 receptors we have, 4 of them are occupied by our insurmountable antagonist. And that means that only 6 receptors are left for your agonist to bind to, instead of the original 10. And no matter how much agonist you add, you're still limited to just 6 sites, because these 4 have been permanently disabled. Because remember, these antagonists are permanently bound to these receptors. No matter how much agonist you add, they won't leave. They're very tightly permanently bound, or at least for a very, very long time. They don't leave very easily. So that means that the number of receptors is basically decreased from 10 to 6. So the decrease in the number of receptors likely means that the total possible cellular response is decreased. And this makes sense because the collective response of just 6 receptors is probably less than the collective response of 10 receptors. So by basically eliminating some of your receptors, the total possible response is decreased. So maybe before, let's say you didn't have your insurmountable antagonist, meaning you had all 10 receptors available, then your agonist could come and bind to them and produce a lot of response. But now that you add your insurmountable antagonist to come and block some of these receptors and decrease the number of basically available receptors that you have, no matter how much agonist you add, you're still limited to a lower number of receptors, so the total possible response is likely decreased. So if we look at the dose-response curves for insurmountable antagonism, they might look something a bit like this. So your original curve, if you add agonist without any antagonist, it will, again, look something like this. So as you add agonist, your response starts to increase, and then increases faster and faster, and then it plateaus. So this is like your max possible response levels. But then this dose-response curve, this represents when you add an agonist along with some insurmountable antagonist. So remember, this insurmountable antagonist is going to block some of your orthosteric sites permanently, meaning that the total number of receptors is effectively decreased no matter how much agonist you add. You cannot overcome this type of antagonism. So no matter how much agonist you add, you will never be able to get back to your original response levels because some of your receptors are permanently disabled. And so your maximum possible response levels are lowered. That's why the plateau for your yellow dose response curve, which is agonist plus insurmountable antagonist, the plateau, which is the final response level, is significantly lower than if you have no antagonist. So whereas with surmountable antagonism, you're able to get that same response level as long as you add enough agonists. So basically you have a shift rightwards. For insurmountable antagonism, no matter how much you add, you're not able to get that same response level. So the plateau, the final response level, it has a shift downwards. So what this means is that surmountable antagonism usually lowers the potency of your agonist since you need a higher concentration to be able to get that same response. Whereas insurmountable antagonism usually lowers the efficacy since the maximum possible response is lowered. So before we go ahead and close out the video, let's review what we've learned today. So antagonism can be between multiple receptors with opposing responses that counter each other out to create a fine balance. This is physiological antagonism. Then there's pharmacological antagonism, which takes place at just one receptor. Now within pharmacological antagonism, you can have competitive or non-competitive antagonism. Competitive antagonism is when you have an agonist and an antagonist both competing to bind onto the same orthosteric sites. Whereas non-competitive antagonism is when you have an agonist trying to bind to the orthosteric sites and a negative allosteric modulator binding to your separate allosteric site. So because your agonist and your negative allosteric modulator are binding to two separate sites, they're not competing against each other. Therefore, the antagonism is non-competitive. So, competitive antagonism, remember, that's when your agonist and antagonist are competing to bind onto the same orthosteric site. So, if your antagonist is one that can bind to orthosteric sites without permanently getting attached, so it can come and go, then this antagonism is surmountable. By adding more agonist, you can outcompete and overcome this antagonism. Whereas if when your antagonist binds to your orthosteric site, it locks on permanently or at least for a long time so that it can't just come and go easily, then this cannot be overcome no matter how much agonist you add. The max response level, you won't be able to reach it, and so this antagonism is insurmountable. And lastly, this is how the dose-response curves primarily differ between surmountable and insurmountable antagonism. With surmountable antagonism, you're able to overcome your antagonism by adding enough agonist, and so there's a rightward shift and a decrease in potency. Whereas with insurmountable antagonism, your antagonists are permanently bound, or at least really, really tightly bound to your orthosteric sites. So even if you dump a whole lot of agonist in there, you cannot fully overcome it, and so there will likely be a decrease in efficacy, a decrease in the maximum possible response. Now, it's actually a little bit more complicated than just a rightward shift or a downward shift. There's more to it, but this is the primary main difference. So that basically concludes this video. We've gone through all of these different types of antagonism and broken them down to become more and more specific, and I hope now you have a better general idea of types of antagonists and how they work. So thank you guys so much for watching. I hope you enjoyed the video and found it informative. If there's anything confusing or anything you'd like me to explain further, be sure to drop a comment down below. I'm open to any suggestions and feedback. And if you like the video, please consider liking and subscribing. I'd really appreciate it. And moving forward, we're going to focus on more signaling pathways of some messenger molecules, such as maybe steroids or prostaglandins. So stay tuned for those. But that's all for now. Take care. I'll see you guys next time.